I serve it.
Okay. Well, I got it on. So okay.
Good afternoon.
Good afternoon.
And at this point, Herbert, you must be in on your cell phone, Herbert?
Yeah, we're
Okay.
still driving back to Long Island.
Okay. Okay. A couple more minutes to join us, and then we'll get started. Hi, Jeff.
I'm in the middle of overexpression right now, so I may have to come and go, but I'll let you know if I leave.
All right. Well, we are recording. The recording is on, and I just see that guest one joined. Ashley. Hi, Ashley.
Hi.
Well, let's go ahead and get started. I know that Ashley has to leave early, so I rearranged the agenda a little bit. We're going to start off just reporting out on the antimicrobial resistance boot camp at Rutgers, and I'll give some numbers, and then I'll let Ashley give some impressions, and maybe Rebecca will be able to join us by then as well. So we had 4 students from Cal Poly, had 3 students from Ursinus, plus 1 high school teacher who was a student at Ursinus some time ago. We had 1 student from SUNY Oswego, and we had 3 students from RIT there. In addition, Rebecca from Ursinus, Ashley from Cal Poly, Julia from Oswego, and I were part of the boot camp. Also included in the boot camp were about 10 or 12 students from Rutgers who took it for credit. In addition to that, there were 2 or 3 faculty from Rutgers who are part of the protein data bank, plus another 3 or 4 people from the protein data bank who are staff there that participated. And we had people come and give us talks from Princeton, from something called PHRI, which is some kind of institute at Rutgers, Public Health Research Institute or something like that. Anyway, we had a couple people from there to speak to us. So all in all, we had a good group. And we were pretty much going full out from 9 a.m. on Monday morning till 1 o'clock on Friday afternoon. Comments from Ashley or Julia or Rebecca when she appears?
Sorry, I missed the first minute of what you said.
Ashley, did you have any comments about the boot camp you'd like to share?
Yeah, no, I thought Rebecca was trying to talk, and that's why. So first of all, I'll say that I don't know how I developed expectations for what it was going to be like. It wasn't really like what I thought it was going to be like. I wish the students had developed more different bioinformatics and more different computational competencies, like that workshop that they had the first afternoon about Chimera. If they had then had one of those about, Pymol or Promol, or if they had then had one of those about docking software, I think that the students would have gotten a lot out of that.
Mm-hmm.
So that was my general impression, is that I wish there had been sort of more workshopped training for specific computational skills.
Right. Rather than almost entirely gathering information or presenting information.
Correct.
Mm-hmm. Thoughts, Julia?
I would agree with that. So I'm not sure you can hear me.
We can hear you. It's a little muddled, but we can hear you.
Okay. It told me I needed to download an app in order for this to work. So then I think the same for the training. If they had some more tools, that might have been useful. But like Ashley, I have expectations.
We're getting about every third word from you, Julia, for some reason. Are you on a phone or just on a bad network connection?
I think it must be a bad network.
Can you hear us okay? Oh, Julia left. She'll log back in.
So I also know that this is not the boot camp way. Like, it became clear throughout the week that what they had in mind happening was the students working very intently all day, every day.
Right.
I won't be able to convince undergrads to go to that again. Like, there need to be more networking activities and more social activities.
Yep.
Like, significantly. Like, we should have all taken the train and gone to a Yankees game one night. Like, that's the level of activity that other undergraduate focused conferences that I take my students to participate in.
Good. Good. Are you back with us, Julia?
Yes, I'm back. Is this better?
Much better. Much better.
Okay. So my phone is much better than my computer today. So yeah, I heard what Ashley was just saying about networking, and that was one of the things I wrote in my survey, is that they needed more networking and better focused networking. I think having the dinners together and the lunches together on some of the days was good, but if they really wanted it to be a networking, and if we really want it to be a networking opportunity, we need to give them some things to do rather than just have dinner together. I also thought we needed to get out of the building more.
Right.
We saw the same four walls often. I think what they got out of it, as far as what they learned about antimicrobial resistance, and the presentation at the end was really good. I talked with Vanessa in the car on the trip back, and she really liked the workshop and what she learned in it, and was very interested in everything. But I think the social events could have been improved upon for that.
Mm-hmm. Good. Good. Okay. Well, I mean, we'll have all the feedback from all the groups, and I did ask people to write like a one-page response, and hopefully they'll be honest and courageous and tell us what they're really thinking. But I agree with what you're saying about more. You know, particularly if we'd had clearer expectations for what they were going to deliver on Friday, then they could have been much more focused in their efforts. And realistically, we could have taken half a day out for other activities on one or two occasions and still gotten the work done.
Yeah.
Yep. That's great. That's great. Okay, good. Good. Okay, the next thing I wanted to talk about was course
Can I chime in for a
source.
second?
Sure, go ahead. Yes.
Sorry about that. So I haven't talked to my students in detail, but yesterday, just asking them, how did you like it? Both Katie and Andrew really did seem to like it, and we were going to have a group meeting later this week where they were going to give us a progress report of everything they learned. So next time I can report back anything that they say, if I think there's something important to share. But they both seem to like it, so that's kind of the student perspective very quickly.
Right. I think it was a positive experience, but coming out the other end, it was pretty intense. And coming out the other end, I'm guessing that they all would have input of things that they would like to see changed. I've gotten some really good feedback from people already. So, and I know that people at Rutgers would be delighted to get constructive suggestions on better ways to do this.
Okay, any feedback I get from the students, I'll pass on to.
Sure. Please do. Please do. Great. All right, let's talk about course source. How do people feel about, is it worth our effort to convert our documents to the course source format and to get them in that system, or do we just want to continue where we are? I'm inclined to have us convert to course source, but I know that's an extra layer of work, and we also need to get things ready for faculty. Now, realistically, the faculty modules, I mean, they're written as goals in our next grant, so we do have 3 years to get those done, but I don't want to spend 3 years getting them done. Other people's thoughts?
Does it have to be either or? Might it be possible to simply, in terms of your new efforts, be doing stuff as course source, but basically don't make it the dominant priority that drives out things that have to have higher priority?
That seems reasonable to me. I might even seek to get a student to convert stuff, to convert the PyRX one that Ashley and I have been working on into a core source format, and then we could
Yeah.
just go and edit that and then submit it. So,
I think a compromise would be workable.
well, now that I think of it, I have. Hi, Rebecca.
Hi.
Now that I think of it, I do have a few dollars left in in the grant that's currently we're currently covered by until this Friday. Actually, most of us I think got extensions for another year. I did, and so I do have a few dollars left in there, and I could probably afford to hire a student to work for me 10 hours a week during the fall semester to at least for part of the fall semester to convert these our student modules into core source documents. So I will seek to hire a student to do that. If anybody is anxious to get involved in that, please contact me. But otherwise, I will just hire a student to do that. Right. Somebody's got a bunch of. That's better. All right. So is everybody clear on what we'll do with core source now?
I'm sorry, Rebecca. I was having computer problems, so I didn't hear the beginning. Can you give me a quick update?
Sure. What we're going, rather than having all of us focus on core source, I'm going to use whatever money I have left in our current grant and hire a student this fall to take the modules, convert them to a core source format, and submit them and see how that goes. Somebody's getting a lot of feedback from somebody.
That was me. Sorry.
Okay.
That sounds great, Paul.
Okay. Good. Good. Okay, let's talk about document review. I haven't looked at Asana today, but I continue to get input that folks are working on those. So many thanks. I know that Anya just completed one on SCS Page, that Colette completed one.
Paul, about that was my first time trying Asana, and I clicked on something, and now I don't know how to make it come back. I'm sorry.
Yeah, I did the exact same thing, so it looks like I did something and I didn't.
I was looking for more details, like where is the document, and you click and it disappeared. I couldn't find it.
Okay, so I'm going to share my screen. The document. Okay, so in the project modules, videos, assignments folder, there's a folder called student modules to be reviewed. And, ones that have been reviewed have different names on here under last modified by. So there are still there are 4 that have not been reviewed yet. And so I'd encourage the people who are assigned to do the determination of protein concentration, SDS page, publish, and blast modules to review those this week. And whoever is the primary author on each of these modules to go in and then make the suggested changes to those modules. And I'm going to go into Asana probably sometime tomorrow and add some more assignments on maybe on second review on some of these. And then I'll also create assignments for creating the teacher version of each of these documents. And
whoever's got that background noise, could they maybe mute themselves?
Yeah, I was going to say the same thing. Yeah. All right, and one of the comments that I think we all shared at the boot camp was that if we could find a way to get all of us together in the same place at the same time for 1 week, we could get a lot of stuff done. We could probably get the teacher versions. We could work on stuff in the morning and then talk in the afternoon about what would be done, and then work the next morning. We could probably have the teacher modules done in a week. And so I'm not going to try to orchestrate that for this summer, but what I am going to suggest is that sometime next in the fall, that we all take a look at our schedules and our budgets and try to figure out some way that we can all be in the same place at the same time, or as many of us as possible, or maybe we'd have an Eastern conclave and a Western conclave or something. Because I know it's a long way from San Antonio and from San Luis Obispo to the East Coast, but we're going to try to work on something like that, because I do think having us all in the same place at the same time, well, it was quite honestly, it was pretty terrific just getting to meet Ashley and Julie in person for the first time ever after we've been together on this grant for over 2 years. But I think being in the same place at the same time, we could get a lot done. Okay. The next item on the agenda is the BASIL blog, and I will add something from the boot camp. I'll link on there out to the boot camp, and I'll put that on the blog. But please be thinking of things you'd like to add to that. And if you've got some summer research students who are willing to have their picture on there doing an experiment, or that's part of this project, that would be great. Colette, you sent out an email that one of your students had tracked down some new proteins for the fall, and I think it would be, in addition to discussing that amongst ourselves, that would be a good thing to put on the blog.
Yeah, I had a kid who he volunteered. He was working as a volunteer, and then I realized I could use my last couple $100 and pay him. And he worked for about 6 weeks. Now he's off studying for the MCAT. But for about 6 weeks, he just sat at the computer, and I had made for him a list of, I had searched on PDB for anything that was called unknown protein, dating from your good hits paper, because I wanted things new uploaded since then. Yeah, and then he went through all of them and looked for anything that also said hydrolase or possible hydrolase, and then he did the promol thing to see whether it looked like they had the catalytic triad, and he made me a short list. Then I sent him to DNASU to see which were available from DNASU, and he gave me a list of 7. I think I ordered 5. I sent you that list. Right. And I said that I thought one, Suzanne, I thought one was I had hydrolase, but I was wrong. I ended up not ordering that one. It was 3R09. I can't remember why I didn't order it. But anyway, so it was pretty great just having this kid do nothing but work on this. So
good.
But he's off studying for the MCAT now, so those days are gone.
Well, it's interesting to me the way that we improvise in terms of research. I wanted to mention something else from the antimicrobial resistance bootcamp. I talked with Stephen Burley, who's the director of the PDB, about how we could get, if our data, as our students collect good data on characterizing structures of unknown function, how we can input that. And he suggested that the very best way to do that, the most politically meaningful way for the authors and the Protein Data Bank would be, if we get meaningful data, to actually contact the people who submitted the structure and ask them if, tell them we're characterizing their protein, and ask them if they'd be willing to co-write a manuscript about it, and then take the data from the, that we use in the manuscript and put that in the PDB. And so let's keep an eye on that. If we, if as we start to see meaningful data for some of these structures, there was one structure that was a a DNA repair enzyme, or a hydrolysed DNA repair or something like that. 3L1W?
3L1W, an endonuclease, yeah.
Nuclease, right. And so, I think we should look at that and decide. I'm not sure if we can do this in a lab or not. In the lab, or we need to have separate research students working on this. But decide how we, if we want to approach that or dig deeper on that. If a couple folks want to pursue that, we should do something with that. So in addition to those 5 proteins, Colette, you said there were more than 5 proteins, but there were 5 that you ordered.
And I can't remember if the cutoff was that DNASU didn't have them or not. I don't remember what my criterion was.
Because
I can go back through my notes and see.
If you find that out, because if we could find another, if there are another 10 hydrolases, we could continue those on our campuses. I mean, the list that Colette sent me, did I share that with everyone? I
think you only shared it with me, and then I shared it with Jeff.
Okay, all right. So if what do folks think of what proteins would you like to dig deeper on the proteins we've already done, or to go to new ones? What are people's thoughts?
Well, this is Rebecca. I'm not sure how to dig deeper within the context of a course.
Right.
Do people have ideas there? Especially since there's certain, there's certain learning goals that are attained in kind of the beginning steps, but I'm not sure what the going deeper steps would be.
Question. Well, I mean, one way we could go deeper would be, okay, so we've seen the paranitrophenyl acetate hydrolysis in the yellow color. We could look at other paranitrophenyl esters, but that still wouldn't tell us anything physiological. I'm not sure where to go next with that. Other folks' ideas?
Do we have any docking results that suggest other substrates?
I still haven't ever gotten PyRX to work for me, so not here.
Okay.
I don't remember. I'd have to go back and look.
Right. It doesn't. That. To be honest, my feeling is that that's been the last thing that people have done, and so we haven't really built on that.
We've had a couple of things we found were lipases, and I looked hard for about a week this spring into ways that we could nail down what that meant. I looked into lipase kits. I looked into possible monoacylglycerides, diacylglycerides. All of those seemed like great ideas. We got a new HPLC at St. Mary's. It just seemed like something we might do. I might, put 1 or 2 tables on something we already know to be a lipase and ask them to go further, but we still can't afford all those materials. They're really expensive.
Right. That's the challenge.
I wonder if we should try to pick one most promising protein, and anyone who wants to do breads does breads, try other proteins, but anyone who wants to try to dig deeper and has the right students for it would, try to get one protein to a place where we can share, with the structure and try to publish something. And one thing we could probably do is try to do some kind of mutagenesis. So if we have a biochemical assay and we have the clone, right, we could try to generate some mutants to show kind of effect of mutation on function, which residue in, that active site is important.
And we got to have a good assay before you can start making mutants.
But you
Right.
said like if we have a lipase assay, right? Even if it's not that specific substrate that's physiologically relevant, would it help to say, well, here are the residues that affect the function? There's like halfway, there's no function, there's like sort of weaker function once you have one assay. Or are you saying, Colette, that we don't even have one good assay for it?
Yeah, that's what I'm feeling. Paranitrophenol decanoate just isn't good enough.
Right. So,
This is Rebecca again. I think that we just need to be cautious of the grant that we've got funded and to keep on track to make sure that we're fulfilling everything that we need to do for that. I just don't want us to all of a sudden be like, oh, we want to publish this one thing and then just move away and not get other things completed.
I agree with that. And I really think that my students have been hugely enriched by doing the little bit that we do. Just getting to that one day where you get a yellow color with paranitrophenol decanoate it's really exciting for them. And that's why I'm doing this. I'm doing it to get kids excited, not to get papers. I know I live in a luxurious place where I'm a full professor and I'm probably retiring in the next handful of years. So I realize I'm both a lame duck and a catbird, but that's how I feel about it.
Guys, I got to slip out because the vice chancellor for research is coming to visit my lab, so I have to go talk to the students.
all right.
Bye.
Bye, Ashley.
Bye-bye.
I mean, I don't think that we should preclude anybody or students from doing further work, but I just don't want us to get, as a group, we tend to get excited and distracted. So we need to stay on task.
I totally agree with it, but I don't know if we have time. Like, I'd like to share an observation that's like really puzzling me. And I had winter quarter, I had students in lab purifying proteins. And then in spring quarter, I had like 3 of those students wanted to continue working on like an independent research project. And it was crazy how unproductive they were on their own outside the sort of even loosely structured lab where a tech was preparing a lot of stuff for them. And so I'm still like really puzzled by this scaling of research experiences and what students get in the class versus what they don't. And it seems like being able to like plan experiments and kind of push in some direction rather than like doing what other students are doing is really, I think, tricky. And that's why I'm really interested in, like, if we're saying our students can do real research, ideally we'd get at least some sort of low-hanging publishable unit done to say that that's research, even though, there are other ways to do research. But if we can, as a group, get something that we could say, yeah, our students published, even if it's like minor paper with like 100 co-authors, right?
It's not true of all students, but most students I've encountered, a switch gets thrown when they get in a project that results in a publication with their name on it. And that switch tends not to get thrown back. It's a very, very important step that then gets them to be willing to dig at other things, even if those things don't directly lead to publication.
Well, I think I completely agree with Rebecca's point. We have a grant proposal from NSF, that's been funded. We need to be true to the course there. And so the question, for example, the question about what the actual function is less of a concern to NSF than the question that Anya raised about taking students from a more loosely structured lab setting to a completely free lab setting where they were lost, where they were foundering. And so those are important things for us to think about. Now, one possibility here is, I know that last last year at RIT, Suzanne focused on on the Nudix hydrolases, because that's her area of research. And so she found some structures of unknown function that were tentative Nudix hydrolases and characterized them with the substrates for them. If you, Colette, wanted to take one or two groups and just had them dig deeper on the hydrolases, or if Ashley decides to really go after PyRX to get some real docking results, it's possible for each of us on our campuses to do separate things in our labs, as long as we're staying with the general course, and this will actually increase the breadth of our story, which is to say, here's a framework, and once you're accustomed to the framework, you can start building on the branches.
Since you brought that up, I was going to jump in, but I'll jump in now. So Paul's right. I focused on enzymes that are in a family that I know. So, and then the substrates are expensive, like Colette was saying, but we went ahead and bought them just for a very specific family. And since I've been working on the field, on the family for like 20 years, I knew like a half a dozen like good like potential hits. And so in the lab, the students did all the procedural stuff that everybody else has done, the bioinformatics, and then expression, purification, and the enzyme assays. But the purification of the enzyme wasn't perfect, and we didn't have a lot of it. And the enzymology was the precursory, can we find activity? And all the students did. But then you really can't do the next step in the teaching lab as easily. So what Jeff and I are doing this summer, we're working with Katie, who was a student in that fall class, and overexpressing a larger amount, purifying the enzyme to purity, or visual purity, and then doing the enzymology much better. And then a paper on that can include all the students that were in that lab, because they discovered an activity, but then like Katie would be a much higher author. And if they don't finish it, if she doesn't finish this summer, then maybe another student or two. And so that way you can do everything that the NSF grant says in the teaching lab, but then people can take it into the research labs. And so what I had said like a year or so ago was, if everybody has enzymes that they've been studying for their career, they could go in and work on enzymes within that family. They'd be more likely to find activity and more likely for it to kind of mesh with the research that they're already carrying out. So that's what we've been trying to do.
Well, I'm going to propose something. I'm going to propose that for the next time we meet, which I think would be 2 weeks from today, that everybody think about what you're going to do this coming year in the lab, and that can anything's available to you. You could continue working on structures we've done before. You could choose to do a more focused aspect on some structures or others, but give that some thought, and then when we meet, as I said, 2 weeks from today, then we'll talk about that. So that'll be on the agenda for 2 weeks from today. Okay?
That's good.
Several people have asked me about the annual report, and our annual report is due, I think, 90 days after June 30th. And so I'm going to get started on that, and I'll get a framework put together on that. Once I have that up, I'll put it in our Google Docs folder, identify a common section, and then a unique sections for each campus where people can report out and get to work on that. So I will get that started.
Remind us when it's due.
I believe it's due 90 days after June 30th. So whatever that, September 30th. I would like to get it done before, by the end of July. So let's say the due date on this is the end of July. I sent out an article called Activity-Based Protein Profiling from Enzyme Chemistry to Proteomic Chemistry. As you know, one of the goals in our new grant is to expand the number of classes of enzymes we can attack or we can study in this lab setting. And I did take some time to read through that article today. And one of the things that struck me as I read the article was the point they made is that over half of the serine hydrolases in humans do not have a physiologically identified substrate. Or maybe it was just half of the hydrolases, because they talk about metallohydrolases and cysteine hydrolases as well. I thought that was interesting. Have other folks had a chance? I believe some of you actually also read that article, and could I get your thoughts on that?
I was busy hanging out with cool people at Rutgers, so I did not read it.
Well, there's much to be said for that.
So I read it, and no, I'd rather listen to everyone else talk about it. It just looked super expensive.
I was going to say, I read it and I wasn't completely certain that it was pertinent to what we're trying to do, because it's a very high throughput thing. So it reminded me of like the Enzyme Function Initiative with these big research labs that are all working together, kind of like the Structural Genomics Initiative. It seemed like the kind of thing that would be done under like one of these big like initiatives, these big clusters. So that's my opinion.
Other comments? I think I saw that a couple other folks had read the article. You know what? I think Jeff read it, and he told me he's going to be. Jeff?
I also read it. I thought it was very interesting, but I guess I can reiterate what Colette and Suzanne just said, that it seems expensive. And also, I didn't see how it would be relevant to us because, like, so if you wanted a method to basically to isolate or to pull down or to see how many serine hydrolases are in a human cell, this is what you would go to. But if you want to analyze just one serine hydrolase and try to characterize it, this is not the method for us.
As I was reading it, I had some thoughts. One of the thoughts is that unless somebody finds a benefactor, we're never going to be able to do the LC-MS stuff they talked about. But that gel electrophoresis is something we all do on our campuses. So that was, but another thought was, we're not doing pull-down assays from entire organisms. We're looking at specific plasmids that express one protein. And so what I'm thinking is, if we could identify, they talk about the fact that there are probes, and the fellow that I met told me that his colleague at Scripps, which I think is Benjamin Kravitz, has something like 10 or 12 simple molecules that are relatively inexpensive that you could use to test things. So my thinking is that if we just took, in our Good Hits manuscript, we had 10 or 12 or serine hydrolases, which we've gone after, but there were some other ones that were in other classes that we haven't pursued. But if, for example, there was a probe molecule that was good for kinases, or one that was good for transferases or glycosidases, or things like that, we could have people one possible thing we could do one year with this project would be to just bring in a variety of plasmids, express them, and then have student groups try basically to purify the protein and see if they can get this fluorescent tag or biotinylated tag or whatever to bind to the enzyme of interest. I mean, it would be a different technology, and so I think we could do it relatively inexpensively. I mean, I've done some stuff with biotinylated tags and immunoassays and so forth that we could find an antibody against streptavidin or something like that, or get an enzyme labeled streptavidin and use that as the tag to indicate that a label had stuck to a protein. At this point, I'm thinking of this as one possible way to expand the number of enzyme classes that we're studying. I'm not hearing a lot of rah-rah in response to my suggestion.
Actually,
You've switched from.
Go ahead, Colette.
You've switched from the substrates being expensive to having to spend a lot of money on all the plasmids.
Well, the plasmids are $35 each.
Pay 43, and you buy 20 of them, and you've just spent $860.
Well, you're right. You're right. So I need to do my homework and get a plan in place. But if I can find a way to get a plan in place for this, I mean, one of the objectives for our next grant over the next 3 years is going to be to expand some, to find a way to study some additional classes. And so, I guess what I'll say is I will attempt to connect with Benjamin Kravitz at Scripps about this, explain to him what we're doing, and see if he has any suggestions. I also found I took that manuscript and then did a citation search of articles that had cited that manuscript, and found there were hundreds of others that had cited the manuscript, but I found several that were reviews that looked promising. So I will keep digging on this while you all are teaching in the lab this coming year. I will be exploring these possibilities.
I have an idea.
Go ahead, Suzanne.
So Karen Allen has, or at least had, this high throughput sort of substrate array. I don't know if it was just for HAD superfamily or if it was for other superfamilies. I don't know if she had more than one array.
Okay.
And you could send her enzymes and they would put it through the array to see what was a substrate hit. And with the only, if you publish this, I don't even think it was authorship. I think it was acknowledgement. But I mean, even if it was authorship, that would be fine. And so it would be a fairly cheap way to screen a lot of substrates if you had an enzyme in any of the families that she could screen. I don't know if there's anybody else that does these sorts of things, but she does it as part of that enzyme function initiative. And I will say I sent some enzymes for research. I think there were two or three I sent, and her hits did not match my hits. That was the only negative. And I haven't had the time to really sort out to see why our hits weren't the same. It might be something to do with like high backgrounds, and I also have to look and see if there were just some false negatives, but if she did actually at least get the same positive hits I did. If that's the case, then basically what they get is hits. Those few things could be bought and tried at the home institution. So it's another way of thinking about doing it. And Colette, in the email exchange that we had yesterday, said that she knows Karen Allen and Deborah Dunaway Mariano. So just a thought.
Yeah, I think both of them would be interested in helping us. I'm not sure that that's in line with what we, I don't know, in line with what we want for our students.
I think that's a really good, like, if we could get someone to do their, massively parallel scale things where they give us some hits and then we follow up with small student groups on potential hits and get a little more detail, the tedious work that people do in these large scale screens don't want to do, that would be a good sort of model for collaborating with them.
Colette and Suzanne, would you be comfortable talking with Karen about this? Telling her about our project and telling her what we're trying to do.
I'll see if she's in Boston when I'm there this next month.
Okay, excellent. That would be great. One of the things that Stephen Burley told me when we were down at Rutgers is that the EFI is no longer funded, and so
that's a shame.
That was a collaboration of 11 PIs on 7 different campuses. It was a big bucks organization, and so he suggested that I contact 2 of them. One named John Girl. Do you know John, Suzanne?
He was a postdoc advisor of mine.
Okay, all right. And another one is somebody he mentioned at Texas whose name I've forgotten.
Frank Rochelle at A&M.
Yeah, that's the one. Do you know
a friend
Frank?
of mine? I know him very well.
All right. So, where to go with this? Well, tell you what. For the first step, Colette, why don't you see if you can talk to Karen when you're in Boston, and then we will. We clearly this summer we've got 6 more weeks left, and I suspect all of us have some vacation in those 6 weeks. But this summer we need to get the student documents reviewed, and we need to get the instructor documents, at least a first draft on each of those. Those are really the priorities for this summer. And so I guess I will bring our conversation to a close with that idea. I will be on Asana sending out assignments and asking people to keep up with that stuff. I guess one of my questions is it reasonable to do this? I mean, are people able to do this, or is this too much?
I was just going to say, I think if people are focused on things that they know well, that might work.
Yeah, my problem with that is my enzyme family is really small. There are only 4 members.
Okay.
Yeah, and I looked for unknown flavor proteins because I have that skill set, but there weren't many. The flavin field is mature, let us say. Surely there are flavor proteins to be discovered, but I was astonished.
Right. One of the things that I've thought about doing, another idea of mine would be to use PyRX in this way, to come up with a set of ligands that bind substrates for that are prototypical substrates for different enzyme classes, and take those substrates and run a series of structures against those classes. So characterize each of these structures of unknown function against a set of 10 or 12 or 15 prototypical substrates that we think would apply to the different enzyme classes. And I'm just thinking out loud of ways that we can expand the number of enzymes we're studying.
Paul, I missed the beginning, but was the workshop at all helpful? Like, I think in terms of finding projects students get excited about, if there was something that we're doing that would contribute towards, combating antibiotic resistance, right? Students would get really excited about that because it becomes meaningful. Was there something in the workshop that might point, like, because then we're just looking, how do you inhibit a particular enzyme, right? And I would think that that might be in some ways a little easier because you don't need biological function. You're looking for an assay and then the inhibitory stuff, which would go with teaching biochem really well.
We didn't really talk about this. I mean, we learned a lot about this. One thing I thought of is that we could create, for example, some motifs in promol and compare structures in the, structures of unknown function from the PDB against motifs for beta lactamases or for penicillin binding proteins, or for some of the aminoglycoside modifying proteins. We could certainly search the structures of unknown function for those traits. We have not done that in the past.
Mm.
All right, so moving forward from here, let's plan to meet again 2 weeks from today. I just realized I signed up for a workshop, but I'll move some things around, or I may ask people, would Monday the 10th, I'll send an email out to ask if people can move. If we can move most of us, then we'll move. If we can't, then we'll just go with that. So we'll meet 2 weeks from today. At that time, we will talk about. I'd like each person to share about what you're going to teach in the upcoming year in the lab course, how you're going to approach it, the structures you want to study, and so forth. And I'm sure I'll have some other agenda items by then. We'll also talk about where things stand. I mean, by that point, I'd like to see that everybody has reviewed the assigned student modules, and that the primary author on each of those modules has made the changes. Mm-hmm. So we'll be talking about that next time, and then talking about getting the instructor modules going.
Sounds good.
The other thought, I must say, I feel like we're all kind of tired. And I know that when we do this during the academic year, it can be exhausting. And so I don't want to burn people out. I don't want people calling me up in October saying, Paul, it's been a good run, but I can't take this anymore. I want to find out a way, sustainable means not only sustainable in terms of how much it costs in terms of money, but sustainable in terms of how much it costs in terms of our lives. And I don't want to burn people out. I appreciated Colette's comment that she's doing this to watch students discover. Please, maybe this isn't the proper venue for this discussion, but please send me your thoughts and comments on what are reasonable expectations. What do you reasonably think you can complete? What do you want to do on this project this summer? I mean, if everybody writes back and says, let's take July off, then we'll do that. At this point, we could do that and come back and hit it hard in August. If let me know what you're thinking.
Okay.
Okay.
Yeah.
Oh, and I see that Arthur Sikora just joined us. Hello, Arthur. Arthur, can you hear me?
Yes, I can. Can you hear me?
Yes. Hi. Let me introduce Arthur to everybody. Arthur is a professor at, what's your university again?
It's LaGrange College.
LaGrange College. And he was actually at, is it the University of Virginia where you're doing some characterization of unknown proteins?
Yeah, that's right. When I was a graduate student, I TA'd for a biochemistry lab led by Dr. Linda Columbus, and she does similar things with thermatoga proteins where the students start with a putatively categorized protein and then work up to getting some information on the activity of that protein.
All right, and so Arthur is planning to implement some or all of the BASIL modules this fall or this coming year in one of his courses. I must have misinformed you, Arthur. We're actually just wrapping up our conversation.
Oh, I thought it said 2.
It probably did. I'm sorry. I have recorded the conversation, so you can hear our conversation.
Okay.
And so, if we can, I don't think I have anything scheduled right now, so I can certainly stick around and chat with you for a few minutes. But I guess what I'm going to do is I'm going to say good day to folks, wish them a pleasant afternoon, and enjoy the 4th of July. And I'm going to turn the recording off, and then I'll just chat with Arthur for a little bit. You're welcome to stick around if you wish to.
Bye all.
Bye.
Bye.